Hi, everyone. Hi, everyone online. Hi, I'm happy today to introduce Dr. Wyatt Shields for today's Cancer Center Symposium. Wyatt started out by getting his Bachelor of Science in Biomedical Engineering at UVA, and then he earned a PhD in Biomedical Engineering again from Duke, where he worked with Dr. Gabriel Lopez on acoustic and magnetic isolation and analysis of cells in microfluidic platforms. Next, he did a mini but productive postdoc with Dr. Velev and Zoucher at the North Carolina State University, followed by a postdoc with Dr. Mitra Gotri at Harvard, where he's thought about, among other things, immunotherapies for cancer, biosensing, nanomaterials, acoustic fluidics, and cytokine backpacks for immune cells. Wyatt joined the faculty at CU Boulder as an assistant professor in 2020 during the pandemic in the Department of Chemical and Biological Engineering and the Medical Engineering program. So shortly after this, he joined the Cancer Center's Tumor Host Interactions program, which is where I met him. Today, he will be talking about engineering strategies for reprogramming the tumor microenvironment. Wyatt, thank you for coming to speak to us today, and I'm looking forward to your talk.
Thank you, Dr. Slansky. I'm going to switch over to this microphone here so I can walk around so I'm not tethered. Give me 1 second. Okay, does this sound okay? Perfect. I'll lower it just a little bit. Okay, so thank you all for allowing me to come and talk with you today. Is it echoey? No? Okay, perfect. Thank you for letting me come and talk to you all today. I'm really excited to be here, not too far away. So as Dr. Slansky mentioned, I'm an assistant professor at CU Boulder. Typically when I give these sorts of talks, I like to show a picture of our campus and try to sell people on the idea of how amazing Boulder is. I think I can skip that for the group here today. But what I will say is if you've not come and visited our campus, I highly encourage that you do so, particularly come and visit engineering. There's a lot of fun things happening on our campus. So as Dr. Slansky mentioned, I'm a new assistant professor, and one of the fun things that you get to think about when you start a research group is, what do you want to be known for? What do you want your group to be known for? And so there's many ways in which you can address this question, and what we thought would be interesting would be to establish sort of a working vision statement. So the working vision statement that I have for my group, and again, it's a work in progress, is that we are interested in the rational design of materials, particularly materials that are active or responsive, and I'll talk about what that means in a little bit, to address major challenges in biosensing and drug delivery. And the way in which we do this is through particle engineering. So my group's expertise, if you will, is in designing different types of particles that are tunable or responsive in different ways, that have properties that we can exploit for specific applications. So these are just some snapshots of things that we've done in the past. I'm not going to go into all of the details today. What I want to focus on is in this title here, engineering strategies for reprogramming the tumor immune microenvironment. So this is my outline for my talk today. I want to start by just giving sort of an introduction about some of the challenges in drug delivery and drug transport from an engineering perspective. And we're going to be talking about this through the lens of nanoparticles. Secondly, I'm going to talk about some of the work that we've done in the last 2 or 3 years, where we combine nanomaterials with immune cells to improve drug delivery. And there's sort of two broad strategies that we have for doing this. One is by engineering particles that can attach to the surfaces of immune cells, and I'll talk about why that's important. And the other is by engineering particles that can be internalized and transported by immune cells. And then finally, because my group is relatively new, I wanted to spend just a few minutes talking about some of the ideas that we have that are currently ongoing. These are new projects that are not yet published. They're actually just sort of getting off of the ground. But I just wanted to sort of share some of the things that we're thinking about, because I think this is sort of a perfect group to get some feedback from. So this is where I want to begin my story today, and I want to talk about nanoparticles and their use in cancer therapy. So nanoparticles are sort of ubiquitous in medicine. They've been used for a number of different indications. And the reason is quite simple. For all these different therapeutics, small molecules, which are often hydrophobic, they're difficult to formulate. Nanoparticles can formulate those particles and allow for facile administration to the body. Peptides, which can denature, degrade, or can be cleaved. Nanoparticles can be used to stabilize those peptides, same with antibodies. And then when you have things like genes which have really high charge densities, nanoparticles can formulate those quite effectively and allow for improved delivery inside of the body. So there's a number of different advantages of why we might be interested in nanoparticles. And if you look at nanoparticles in the context of cancer treatment, you'll see there's been some pretty steady growth for the last 20 years or so. So this is a Web of Science search looking for nanoparticles in cancer. And I think this is really interesting, but there is sort of a grand challenge that hasn't been addressed. It's sort of an elephant in the room, and it's where I want to start my talk today. And that is this idea of systemic delivery. And the punchline is basically nanoparticles fail to infiltrate tumors effectively. They fail to infiltrate solid tumors. So this is an image or a graph from a review paper that I love. It's from Warren Chan's group at Toronto. It was published in 2016. And if you haven't read it yet, I highly encourage you to check it out. It's really, really interesting. What the authors did was they looked at over 200 papers in the literature that involved nanoparticles in cancer treatment. And in particular, they looked at nanoparticles that vary in size, that vary in shape, that have been modified in different ways to bind to different cancer antigens, or tumor-associated antigens, I should say. Different types of cancer, different stages of cancer, and so on. And what they found is that the median efficiency of delivery was 0.7% across the board, which is abysmal, right? You know, a lot of people look to claim, oh, these nanoparticles are super, they're targeted, they're really effective. Maybe they're effective, but I wouldn't say that they're really targeted. And so one of the consequences that's overlooked is this idea of toxicity that isn't adequately reported in a lot of these papers. The last thing I'll note on this graph before I move on is that we love to think that as you put more money into a problem that we sort of evolve or progress, right? And so you would like to think that over the course of 10 or 11 years, by dumping more and more money and doing more and more research, that we would get better at targeting. But what you see here is over the course of 10 years or 11, over the course of 11 years, we really have not gotten any better in terms of targeting these solid tumors. So what I want to challenge us to think about is how can we address this? This sort of suggests that there's a fundamental limit. There's a fundamental mass transport problem here. And conveniently, immune cells have the advantage of millions of years of evolution. Immune cells can target inflammatory signals in the body, and they can respond in a really specific and a really effective way. The efficiency is quite dependent on a number of different factors, and in fact, what do I want to say here? The exact efficiency of targeting is very dependent on the type of tumor, the type of immune cell, the progression, a lot of different sort of factors. So it's hard to pin a specific number on it. But this is from one particular paper that looked at monocytes and targeting 4T1 tumors in mice. And they found that around 7% of these immune cells of monocytes will infiltrate solid tumors. So whenever you see something that's 10 times more effective in the literature, that's something to really get excited about. And so that's where I want to begin today. And I want to start with this question. And the question is, can we use immune cells as next generation delivery vehicles? The idea is we want to sort of take a lot of the engineering and the chemistry that's been developed or used to make nanoparticles, but then integrate that in with immune cells to have a more effective system. So this is sort of the new paradigm that I'm presenting here today. I'm going to focus here on the right-hand side of the slide. What we want to do is, I'm going to sort of pitch two ways to think about this. One is to design nanoparticles that can attach to the surfaces of immune cells. And I'm going to talk about the different design rules that are required to facilitate this, this interaction. And the other is to load nanoparticles within immune cells. And then I'm going to talk about the corresponding design rules for that as well. And so for both of these, we have to really think about this from a materials perspective, from a chemistry perspective, and that's really what I want to focus on today. But before we begin, I do want to give one example. This is from a paper that was published earlier this year, and it's not an immune cell per se, of course, it's a red blood cell, so it's a very different system, but it illustrates the concept, I think, quite beautifully of the potential that I'm describing. So what the authors did was they loaded CXCL10 into PLGA nanoparticles, and they modified the surfaces of those particles with an antibody, anti-ICAM1, which is expressed on endothelial cells. And they injected this into the lateral tail vein of mice, and the idea is that once these cells get into the capillary bed of the lungs, they experience higher shear stresses, and those shear stresses essentially pull off these particles, and the particles then can deposit themselves inside of the lungs. And so you can see here within 20 minutes, it might be a little hard to see on the screen, but if you look at the deposition of lungs, and if you look at their EASY experimental group, this is their platform, you can see that the efficiency of delivery in the lungs is quite high. And you could argue that that's partially due to where they inject it, but nonetheless, they show that this is really quite effective for treating lung metastases. And so I think this just is one example, again, that illustrates how we could think about combining materials and cells. And the reason why I like immune cells in particular instead of red blood cells is really for two reasons. The first of which is that immune cells express chemokine receptors that allow them to respond to specific signals from different types of tumors. That provides an opportunity for engineering. What if we could select immune cells that are auspicious for a certain type of cancer? The second is that immune cells could participate in tumor immune activity. Since you have this slow release capability from these particles, you can actually use these immune cells not just as delivery vehicles, but as workhorses that can contribute to cancer treatment. So in particular, I'm going to talk a bit about macrophages as some of the, as the vehicle that we've used in my sort of recent work. And we like macrophages for two reasons. The first of which is they infiltrate tumors fairly efficiently. The efficiency, again, ranges, but 4 to 8% is not an unusual number in terms of injected dose. And the second reason is that macrophages are highly phenotypically plastic. They're one of the most phenotypically plastic cells of the hematopoietic system. And so this means that they can be sort of reprogrammed to have a certain functional phenotype, which allows us to treat tumors in an antigen agnostic way. And so that's the one reason we're really excited about this. We don't have to have knowledge a priori of what is the molecular composition of a certain tumor. We can try to use these immune cells and really sort of leverage this innate immune system to fight these tumors. So this is not necessarily a new idea. There were some clinical trials in the 1990s that used, quote, macrophage-activated killers. The goal was that you would take macrophages ex vivo and you would polarize them toward an M1-like phenotype using interferon gamma. You would inject those cells intravenously, and they would localize into a tumor, and they would release cytokines that would effectively generate a cascading event where you could reprogram these tumor-associated macrophages. So that was the intention. But what ended up happening in reality is that due to the highly immunosuppressive tumor microenvironment, these immune cells would quickly lose their functional phenotypes. And so you can see the outcomes of these different trials here. It was quite disappointing. So the question that we're asking is, how can we maintain phenotypes in a highly suppressive tumor immune microenvironment? And we want to integrate this slow release aspect. So one of the advantages, again, of nanoparticles is that we can play with different chemistries, and we can, increase or decrease the rate of drug release in a way that is tractable, that is totally up to us, if you will. And so what if we can sort of take that concept and apply it to this problem? The challenge, of course, is that macrophages in particular are amazing phagocytes. So I think it goes without saying to this audience, but I'll show this video anyway because I just love it. Here is a macrophage. This is a human macrophage, and I think these are sheep red blood cells. And you can just see these macrophages love to eat everything in its environment. So it's just a fun little video to watch. I would love to do this experiment in my lab just for fun. So if you have a particle and you want to introduce it to a macrophage, odds are it gets internalized. And so where am I going with this? The question is, how can we evade cellular phagocytosis? Well, there's a really, really amazing study that I read when I was in graduate school about these particles that were non-spherical. In fact, these are ellipsoidal particles. If you're interested in how they're fabricated, I'd be happy to talk to you about it. It's essentially a method called particle stretching. So you take a sphere and you stretch it along one of its axes and you make an ellipsoid. And what the authors found is that the angle of this ellipsoid with respect to the cell as it encounters the cell, or vice versa as the cell encounters it, would dictate whether or not it's internalized. So on this x-axis, you can see basically that angle I just described, and on the y-axis, you can see internalization velocity. And effectively, what the authors found is that at a certain critical velocity, namely when a particle is orthogonal to a cell, so it's sideways, if you will, these macrophages just cannot internalize these particles. They get frustrated, and so the particle will end up just resting on the surface, as you see in image B here. Whereas in image C, if it's oriented the opposite way, it's internalized quite readily. So this is the first innovation I want to talk about. We call it backpacks because it's really easy to remember. Backpacks help us to evade phagocytosis, and we can resist those bad influences from the tumor microenvironment quite easily. It's sort of a play on words here, but I think it illustrates the basic idea. What we want to do is we want to take these backpacks, engineer them to slowly release cytokines that allow the cells to which they're bound to maintain a certain phenotype. So that's the goal. And just to kind of, I guess, illustrate this in a different way, this is here, this image shows effectively what the clinical trials that I showed you on the last slide, what they tried to do. They took interferon gamma, they took macrophages, polarized them toward an M1 phenotype, and once they get into this tumor, they lose their M1 phenotype. Whereas if we have these backpacks and we bind them to cells, once they infiltrate these solid tumors, these backpacks allow the cell to maintain its phenotype and even potentiate a pro-inflammatory or anti-cancer phenotype of tumor-associated macrophages. So that's the basic idea, and I'm going to talk about how we built this system here over the next couple slides. Again, I love engineering particles, so this is what we do with a lot of our time is think about fabrication methods. I'm going to kind of go through this quickly because I realize that's not, may not be super interesting to this audience, but again, if you're interested in this, we're happy to talk more about it later. But the basic idea is we're using soft lithography to print these biocompatible polymers in different layers onto a surface. So we deposit these different layers that comprise our backpack. And then once we have the final layers built, we basically flip the surface over, we press it down onto a released polymer, polyvinyl alcohol, and then we're left with these disc-shaped particles. We call these our backpacks. And since it's on a PVA film or polyvinyl alcohol film, once we add water to this printed layer or this printed array, within about a minute or 2 minutes, you can see our backpacks come into solution, and then we can mix these with our cells. Okay, so that's basically how we make them. What we did was we took, these are all animal studies. We took bone marrow from bulb C mice. We cultured it in MCSF for 7 to 10 days. So we would differentiate into macrophages, and in parallel with that process, we would fabricate our backpacks. And at the end, once both are ready, if you get your timing right, you mix them together for about an hour and a half. That facilitates binding. And then after that hour and a half, we wash off anything that's not bound, and then we collect our cell backpack complexes. So this is what it looks like when you follow those different steps. Here you can see our backpacks are colored pink or red, and that's due to rhodamine, which is conjugated to the PLGA polymer, which comprises the majority of the backpack. The first thing that we did once we made this was we looked at the internalization of these particles. And so on the x-axis here, you see time in days. And on the y-axis, this is the percent. It's a little confusing. It's the percent of cell-associated particles that are not internalized. So surface, so you don't have a free particle. It's just particle that's associated with a cell that's not completely internalized. So what you want to see is 100% or something close to it. And what we see here is in the case of blue, that's our interferon gamma containing backpacks. We see after the course of even 5 days, the majority of these backpacks remain surface bound. Whereas if you have spheres of the same effective volume or a slightly larger effective volume, they're internalized quite rapidly. After 2 days, around 90–95% are internalized. So that just, again, illustrates that this idea of shape is so simple, but yet it's so profound in terms of the phagocytic fate of these particles. A little bit more just about the construction or the composition of these backpacks. We built these layer by layer assemblies. This is an electrostatic assembly of different polymers that will help improve the attachment of these backpacks to cells. So this is a hyaluronic acid layer, which has an affinity for CD44. So we have this as a top layer onto our backpacks. We have PLGA. This is a biodegradable polymer that's used in a lot of drug delivery applications. This gives our backpack some structural integrity. We have polyvinyl alcohol. This is our hydrophilic layer. I tried for 6 months to put cytokines into PLGA, and other people have shown you can do it, but I just could not get it to work personally. So I figured out if you integrate this PVA layer inside, you can actually stabilize the cytokines that way. So it turns out that works pretty well too. And then finally, we have another PLGA layer, and that just, again, provides some structural integrity. We characterize the stiffness and dimensions with AFM. And here's the release of interferon gamma over the course of tens of hours. And what we found is that with these backpacks, we can load around 80 to 100 femtograms of interferon gamma per particle. So once we made these backpacks and we showed that we can stabilize interferon gamma inside of them, then we wanted to look at their effects on immune cells. And again, these are the bone marrow-derived macrophages that I described previously. And we have 3 groups here, first of which is macrophages with free interferon gamma. This sort of recapitulates the clinical trials I mentioned. We have macrophages plus blank backpack. So this is to illustrate or show if there is an effect from the backpack itself without any cytokines present. And then finally, we effectively combine the two. So these are macrophages plus the interferon gamma containing backpacks, and that's shown in blue. And I always make this blue because blue is my favorite color. So we look at 3 different pro-inflammatory cytokines here. We looked at a number of different ones that are in the paper, but I'm just going to show you these 3 for the sake of time. We have INOS, MHC2, and CD80. And what I'm plotting here on the y-axis is the fold change in expression compared to control of no treatment. So compared to baseline, if you will. And what we find is that in the case of INOS and MHC2, you see a fairly strong synergistic effect where the combination of the blank backpack and then the free interferon gamma doesn't quite account for this signal that we're seeing with the interferon gamma containing backpacks. So it's really interesting. To be totally honest, we don't really understand why there's some synergy here, but we see it, and it's really quite remarkable. And I'll point out too that this y-axis is logarithmic. So it's really strong for INOS, MHC2 to a lesser extent. CD80, there was a very slight increase, not very significant, but I'm just including that as well. And this was over the course of 5 days. We can see that these cells are maintaining their phenotypes for that duration. So then what we did was we took our cells in backpacks and we injected them directly into a tumor of mice. And what we did was we inoculated bulb C mice with 4T1 tumors, and then we injected our cell samples directly into the tumor. And the question that we're trying to ask here is, can these cells or can these backpacks allow cells to maintain that pro-inflammatory phenotype even inside of the really immunosuppressive microenvironment of a tumor? So we're looking at the same 3 markers here at the top again, INOS, MHC2, and CD80. And what we see here, these are the 3 groups. I'll walk you through them briefly. These are macrophages that are polarized ex vivo with just free interferon gamma. Group 2 is where we co-inject macrophages with free interferon gamma. And then finally, this 3rd group is where we have the combination. And we do see a significant increase in INOS, MHC2, and CD80. Interestingly, the effects of MHC2 and CD80, the relative fold change was fairly similar. INOS, it was not nearly as much. Again, we're not totally sure why, but nonetheless, we did see significance compared to the other groups. And this is all fold change in expression compared to saline controls. Next, we decided to ask a different question, and that is not about the cells that we're injecting, but the endogenous macrophages, so the tumor-associated macrophages. And the reason that, or the way that we were able to distinguish these two populations was because we labeled our injected macrophages with VivoTrack, which is a really stable dye. It doesn't leach out very easily. And so we could essentially phenotype our endogenous TAMs separately from our injected macrophages. And we see there is an effect. It's not as strong as the cells, of course, that we injected, but we do see, compared to saline, that there is a significant increase in INOS and MHC2, not so much for the case of CD80. One of the last things that we looked at in the study was, okay, how does this affect tumor growth? Since we've done all this work, we just want to see, does it have an effect on the growth of these tumors? So what I'm showing you here in black and green, black is saline injected right into the tumor. Green is macrophages plus free interferon gamma. We have 2 injection days, days 9 and 14. What we see here is that tumor growth are almost indistinguishable. From a statistical perspective, they're identical. But when you take interferon gamma and you formulate it into the backpacks, we can cut this tumor growth in about half. It doesn't completely eradicate tumors, it doesn't revert growth, but it does slow down tumor growth fairly significantly. So this was a result that we were pretty excited about, especially because the amount of interferon gamma that we put into these backpacks, or the total dose was 50 nanograms, which is about 1/100th of the maximum tolerated dose. So I think this is a nice result. We didn't really optimize it any further beyond this, but I think there's some interesting opportunities for additional work. Last thing I'll show on this project is we also looked at lung metastases. We had 4T1 luciferase, so we're able to use IVUS to look at metastatic colonies inside of the lungs. We can see that after 32 days, there is less signal coming from the lungs, so that would suggest that you have fewer metastases. And survival was improved, but not by a lot, but it was improved by a little bit. So that's that. So the idea here is that, and what I want to end on with this project is that backpacks can sustainably regulate macrophage phenotypes. We just looked at interferon gamma. I think there's some really interesting opportunities in the future to look at different drugs and drug combinations, different cytokine combinations. We showed that they can attach to macrophages, they avoid phagocytosis, we can guide phenotypes in vivo, and there's a number of really interesting future directions that we're thinking about, and if you have ideas, I'd love to hear those as well. So where are we? We've talked about surface engineering strategies. I want to spend just a few minutes talking about a different angle, and that is, can we design particles that can be internalized and can that be used for cancer therapy? And admittedly, this is not an immune reprogramming project, but I'm going to include it anyway because I think it has implications for the same. So here we're loading nanoparticles within macrophages, and there's some design rules I want to start off with off the bat, and that is our nanoparticles need to be appetizing. We want our macrophages to eat them, so they need to be appealing from an internalization perspective. Secondly, they need to be durable. They need to withstand a lower pH of the cell lysosome. And finally, most importantly, it can't kill the cell, right? So if you have a high concentration of a drug, let's say a chemo drug, that's going to kill your cell. And then if you inject this in vivo, it's not going to go where you want it to go. So that's obviously a non-starter. So when we started this, we were thinking, okay, well, a hypoxia-activated prodrug would be really interesting here because these drugs increase their activity in a hypoxic environment. So in a low or in a normoxic environment, they're not as quite active, and so cell death is not as much of an issue. And they really sort of turn on when they get deep into the hypoxic regions of tumors. So we're using tierpazamine, which turns into a toxic radical in hypoxia. We want to formulate these into nanoparticles, feed them to macrophages. And basically the idea is that macrophages can help transport it deeper into these tumors compared to free nanoparticles or free tierpazamine. So this is basically the same idea, just shown in a really colorful picture. If you'd like, the idea is that these cells can transport these hypoxia-activated prodrugs deep into this hypoxic region. So this project was led by a good friend of mine and a really brilliant chemist, Michael Evans. And Michael tried to formulate, or first what he did was he would conjugate TPZ to these lipid tails. And then he would try to create basically these fatty balls that would serve as his nanoparticle, and he would feed those to macrophages. But what he found is that those were not internalized very effectively. In fact, try as hard as he may, they just wouldn't be internalized very readily. So here, what you're looking at is an image of tierapazimine with macrophages incubated for a couple hours, and then everything is washed off, and you see not very much is internalized. But if you co-formulate this with PLJ, the same polymer that we use in our backpacks, the internalization goes way, way up, and I'll just show you the data here. If we look at the concentration of particles in the x-axis and the uptake in micrograms per million cells, you can see there's a fairly linear relationship, broadly speaking, which suggests that internalization is not a rate-limiting factor. These cells are readily taking these particles up pretty easily. So that meets the first criteria. They are appetizing. The next one is toxicity. Michael went and did a bunch of different studies where he looked at the concentration of particles, and he looked at the toxicity in normoxia and hypoxia. And what he's interested in, and what we're interested in, is the greatest differential. So you want them to be most viable in normoxia, and you want the viability to be lowest in hypoxia. And basically, if you look at all this data at a fixed concentration of 300 micrograms per mil, you see that there's a clear winner. Tierapazimine C12 has a really high differential in toxicity. So that's the formulation that we went with moving forward. And then when you inject this, or you feed these to macrophages and you inject them systemically, what you see here is if you just have particles alone, so there's no macrophages carrying it. I don't know how well this translates on the lighting or on the screen here, but there are three little arrows that are pointing out these tiny, tiny red dots. So there's some nanoparticles that are getting into the tumor. This is not quantitative, it's very qualitative, but you can see here it's very, very little. Whereas if you have these same particles that are inside of macrophages, the efficiency or the amount of particles that you see in these tumors is significantly increased. So as a monotherapy, then what we did was we looked at tierapazimine nanoparticles inside of macrophages, and we looked at the therapeutic effects. And you can see here that the tumor growth is best. Again, this is a 4T1 model. Tumor growth is most suppressed in the case of the macrophages with the TPZ C12. And it's not shown here, but I think one of the reasons why we thought that this was not as effective as it could be is that this was really good at clearing out the hypoxic areas of the tumor, but the well-oxygenated regions of the tumor, I think, were still fairly intact. And so I think combining this with other therapies that attack those regions would be an interesting direction. So this just kind of summarizes those design rules. We want it appetizing, you want it to be durable, and you want concentrations to be appropriate. And so those are just things to think about. Future directions, we want to look at chemotaxis efficiency versus nanoparticle loading. As you load more and more of these particles, how does that affect the distribution inside of the body? I think that's an open question that needs to be looked at more closely. Combination therapies, pH-sensitive drugs, these are some interesting things that we're thinking about. Okay, so I think that wraps up what I wanted to talk about with numbers 1 and 2. What I want to do with just the last few minutes, maybe the last 10 or 15 minutes, is just talk about some of the new ideas that we have in my lab. As Dr. Slansky mentioned, I started my group January 2020, so we're still getting a lot of projects off the ground. And I think this would just be a really fun opportunity to talk about some of the ideas that we have and just share them with the community. So I have 3 ideas I'm going to show. The first one is this sort of shape change off switch. This is building directly off of the backpack work that I showed you in the first half of my talk. And the challenge here that we're trying to address is this prevalence of adverse events. So immunotherapies, generally speaking, do have some challenges with immune-related adverse events. And it's unfortunate to have to send patients to the ICU after you give them like CAR T cells or systemic cytokine infusions. So what we wanted to do is we want to think about, is there a way that we could use our engineering tools to create an off switch, essentially, that after you administer this into the body, is there a way that we could remotely just turn things off, almost like a switch? And since we have this backpack technology, it actually turns out that maybe it's not so difficult. So what we're working on is the idea here as follows. We have these discs, they remain bound to the surfaces of our cells, and as long as they remain bound to the surfaces, they're releasing these cytokines that are affecting these immune cells. But as soon as toxicity emerges, what we can do is we can trigger a shape change with magnetic hyperthermia. It turns that disc into a sphere. It effectively melts the disc at like 42, 43 C. That temperature is determined by the molecular weight of the PLGA, so that's something that's pretty easy for us to do. And then we can just melt this particle and have it be internalized within just a few minutes. So this is what we're working on currently. These are sort of the 2.0 iteration design of these backpacks. It's fairly similar. It's a single layer design, which makes fabrication on our end a little bit easier. But the big distinction here is that we're incorporating these magnetic nanoparticles. And what that allows us to do is we can apply an AC magnetic field. So it's a really high frequency field, 200 to 400 kilohertz. So basically the field director is oscillating that rapidly. And what that does is it basically it imparts torque on the particles, and then they're moving around really rapidly, and they're generating heat inside of the backpacks. And so as they're moving back and forth, and they're heating up, it basically melts the polymer, and the polymer melts into a sphere. So the open questions that we're asking, and again, this is very sort of early stage, is what are the timescales of shape change, and what are the resultant phenotypes, and are they programmable? Does this actually give us a way to turn off our drug? Stay tuned maybe for the next year, and we'll find out. So that's the first idea. The second idea is probably my most complicated idea, but it's one that I'm really, really excited about. So I'm going to do my best to really break it down, and hopefully it's easy to follow. So the second idea is this acoustic cell preparation system for adoptive cell transfer. I'm going to show you the whole system initially, and I'm going to break down each of the components and kind of walk through what they're doing and why it all comes together, or how it all comes together. So we have these particles, we're loading them with different immunomodulatory drugs, and we're labeling them with cells. We feed this into an acoustic standing wave, and we separate out unbound particles from cell-bound particles. Once we have that, then we inject this into an animal, and then these cells can transport these particles more effectively into a solid tumor than these free nanoparticles could. So there are two things that it turns out that can be used or leveraged to resist phagocytosis. The first is you can engineer shape, which we talked about with the backpacks. The second is elasticity. It turns out if you make particles really, really soft, cells have a hard time internalizing them. Again, we don't know exactly why. It has something to do with actin filament polymerization, but cells just in general have a hard time internalizing them. This is a paper from ACS Nano in 2015. What the author showed is that if you make particles with different moduli, so basically different levels of stiffness, the softer particles would not be internalized quite as easily. So this gives us sort of another dimension to think about when we're designing our particles. So what we want to do is we want to create really, really soft particles, in this case, that can hitch a ride on immune cells. So the idea is, again, we put drugs inside of these particles, we bind them to a macrophage or a monocyte. But the challenge is, if you're thinking about monocytes or cell suspensions, if you have a mixture of cells and particles, the question is, how do you separate out your particles that are bound to cells from those that are not bound? Because remember, if you inject these free particles, they'll accumulate in the liver, spleen, and off-target regions. And so we really want to just infuse particles that are bound to these cells to give us the optimal biodistribution. So how do you do that? I think a lot of folks here are quite familiar with FACS. You probably use it in your research. FACS remains the gold standard for cell purification. This was developed 53 years ago by Fulwiler. And what's amazing is that the technology developed over 50 years ago has remained exactly the same for what's used still today with FACS machines. I won't walk through basically how it works, but if you're interested, we can talk more about it. It is really cool stuff, but it is fairly primitive. It's got a lot of advantages, high fidelity, it's really rapid, but the problem is that it's so massive. It takes up a whole lab bench. It's tedious to run. You have to have all these SOPs, right? And it's not very user-friendly. So one of the things that I worked on, and I have just one slide on this because this is sort of near and dear to me. This is what I did my PhD work on, was Acoustic Fluidics. So Acoustic Fluidics is a really cool way to manipulate particles and cells in small microfluidic devices. So I'm going to show you this really fun video. This is a gentleman, I think from Oak Ridge National Lab. What you see here is you see two bullhorns that are emitting a sound wave, each of them at around 22 kilohertz. So this is right above our audible frequency range. And what they're doing is you have sound waves that are moving in both directions, and you're forming what's called a standing wave. So if you remember from physics, what's happening at a standing wave is that these particles are being trapped at the pressure nodes at each of these points. So what's happening is these are styrofoam balls. They want to fall down to the force of gravity, but this pressure wave is pushing them up. But it's not pushing them up continuously, because otherwise you just see the particles move up all the way to the top. What you're seeing is they move to a stable location and then they rest. That's the pressure node. And it's totally a pressure-driven effect. It's all acoustic fluidic, and it's levitation. So this is truly levitation using sound waves, which is really quite remarkable. What acoustic fluidics does is it takes that concept and it scales it down to a microfluidic device. So this is the nuts and bolts of how we make these devices. I make these in my lab. Currently, we just take some silicon wafers, we etch them using deep reactive ion etching. We fuse a glass lid using anodic bonding. I'm going to skip maybe some of the equations. What I want to really focus on is really on the right side of this slide, and that is, how do particles and cells respond to a standing wave? It turns out most biological cells and particles, if you put them into a standing wave, they're going to move to the pressure node, just like you saw in that video. And the reason is because the pressure is stable in time and in space in that location. So imagine here, this is a microfluidic device. The fluid flow is coming out at you, and so these particles are kind of centered at the focus, and this is a half-wave standing wave, so they're focused here at the center. So the question is, what if you infuse elastomeric particles, these soft particles that I was talking about that are really good at resisting phagocytosis? What if you put those into an acoustic fluidic device? Well, something amazing happens. They move to the pressure antinodes. This was what I worked on for 5 years, and this is really, really cool stuff. And what's really exciting about this is you have a complex mixture of cells and these really soft particles. You turn on a standing wave. What happens is you get this rapid indiscriminate separation, which has the potential for higher purities than magnetic separation. And the reason, because if you're interested, magnetic separation just basically pulls things to the pressure maxima, right? Whereas in acoustophoresis, you move things, some things move in one direction and other things move in a different direction. So you get this discriminate separation, so it gives you higher purities, and it eliminates that issue of entrainment if you ever do MAX in your lab, or magnetic activated cell separation. So now that we've kind of walked through all the different elements, we're bringing it all together. So we have these really, really soft particles. They evade phagocytosis, and they migrate to the pressure antinodes of a standing wave. So what we want to do is we want to take these particles, load them with immunomodulatory drugs, fuse them with these cells, inject them into our acoustic fluidic devices, rapid and indiscriminately separate the bound particles from unbound particles, and then we inject this directly into our mice, or humans eventually, but one step at a time. And the reason why I think this is really, really exciting, and I get a little worked up about it, is because this addresses some of the limitations of CAR-T. We're not using anything that relies on antigens here. This is totally using the innate immune system. We're selecting drugs. This is TMP195. We're also looking at rosiquimod, other types of small molecule drugs that are really stable, easy to formulate. These are drugs that can reprogram immune cells within solid tumors. Secondly, we're using monocytes, sort of off-the-shelf allogeneic cells. So we don't have to isolate, transfect, expand ex vivo. We can bypass all of that. And it also brings down the cost and the time scale. So how do we make these biodegradable particles? This is what we've been doing in my lab for the last year or so. We have this really beautiful class of polymers, PHA. These are naturally produced by a lot of bacteria through fermentation of lipids and sugars. They have a bunch of really interesting properties. But what I'm going to show you here, this is some preliminary data that we've just gotten in my lab. What you see here is that these particles actually do, in fact, migrate to the pressure antinodes, which is exactly what we want to see. And what also is really cool is that if we put in hydrophobic dyes, they encapsulate those dyes really, really well. So that shows that we can put drugs in here that are also really hydrophobic. And so here we have our cells plus NACPs. These are negative acoustic contrast particles. They're moving to the node, and here we see our unbound particles at the antinode. So indeed, the physics of acoustic separation applies here as well. And so now what we want to do is we want to start loading in different drugs into these particles, and we want to administer these and see how they perform inside of the body in terms of biodistribution and tumor immune reprogramming. The last thing I'll talk about with this project, and then I'm going to just show the last one. It's a lot simpler, I promise. Acoustic Fluidics gives you this idea of multi-harmonic scaling. So just like if you're familiar, if you've taken music classes or if you've ever played an instrument, you're familiar with this idea of harmonics, right? Well, acoustics, you can play similar tricks. You can play with the frequencies and you can have multiple parallel channels. And so one thing that we're really excited about doing is creating these microchip devices that have multiple parallel pressure nodes. And the idea is that this allows us to scale up how many particles and cells that we can separate. So if in a single channel you can separate around 100,000 cells per minute, this means that we can do maybe about 1 million cells per minute. So that's one of our design goals for this project. And if we can, then that means we can purify maybe 200 million cells on the order of just a few hours. And so if you think about the timescales of thawing monocytes, performing the binding step, and then doing the acoustic separation, and then doing the infusion, this can all be done in a day. So less than 8 hours, less than 6 hours. And then finally, it's antigen agnostic. So we're thinking this would be a really interesting technology for relapsing and refractory solid tumors. Okay, hopefully that makes sense. If not, I'm happy to answer questions. Here's the third project. This is also using acoustics, but in a very sort of different way. So this is using high intensity focused ultrasound. So instead of a standing wave where things move to a node or an antinode, we're using a traveling wave that has a higher force associated with it or higher pressure. So what we want to do is, this is in collaboration with Andrew Goodwin. He's in my department, Chemical and Biological Engineering. Andrew Goodwin's group has made these really beautiful mesoporous silica nanoparticles. And what he's shown is that if you render the surfaces of these particles hydrophobic, what you can do is then you can backfill these particles with phospholipids. And so those phospholipids effectively protect the particle and allow you to trap in air inside of these pores. And so once you have this particle that's constructed, what you can do is then you can hit it with HIFU, again, high intensity focused ultrasound. And what happens is you cavitate bubbles that are trapped inside. So these bubbles will expand and then they collapse. And upon collapsing, these particles will propel. And they propel really, really fast and really forcefully. And these particles also are really, really small. So if you put them inside of a cell, they puncture through the cell and they actually can swim their way through tissue tens of micrometers, which is really quite fun to watch. So what we want to do is we want to take these mesoporous silica nanoparticles and feed them to macrophages. So again, we want them to be appetizing, so we're going to modify the surfaces with different groups that make them appetizing, and this is sort of the workflow. So we'll have these cells, they'll be loaded with these mesoporous silica nanoparticles. We inject them systemically, they localize to the tumor, and what's really interesting is that we can then just apply HIFU to the tumor region. So only in the tumor region will you have this HIFU, and so only in the tumor region will you have this sort of really disruptive propulsion event occurring. And as these particles propel, what we want to do is we want to load in hydrophobic drugs right at the interface between the mesoporous silica that's hydrophobically modified and the phospholipid layer. So that's a really, really nice layer. We have a lot of surface area. We can load in hydrophobic drugs quite efficiently into that space. So we want to put in drugs that have the potential to potentiate an immune response, specifically with tumor-associated macrophages. Excuse me. So I think this is my last slide. This is just some preliminary data that shows how these particles work. These are not with macrophages, these are just the particles by themselves, and this was all just done in vitro. This is a 4T1 tumor spheroid model. So here you can see the spheroid. We apply HIFU with these mesoporous silica nanoparticles, and you can see it just totally disrupts the entire spheroid. So this is the calcine stain. If you look at the different controls, this is untreated, so there's no HIFU, there's no particles. You can see they're viable. This is what a spheroid looks like when it's dead, so calcine signal's lost. If you only apply HIFU, the spheroid is fine, and if you just have the mesoporous silica, the spheroid is fine. So this illustrates that these particles are really quite powerful, they're really quite effective. You don't need a really high dose in order to administer this really sort of destructive detonation. And they're really good at killing cells. So I think this will be a fun project to play with. All right, good. So this is my conclusion slide as promised. So in conclusion, hopefully I've sort of shown you how we can use engineering strategies to to build different types of particles. I've walked you through some examples where shape plays a role, elasticity, drug loading, composition, surface chemistry. These are all different sort of knobs or levers that we can play with to have functional materials that have some interesting effects that we can take advantage of. We've talked really about antigen agnostic therapies. That's really been my focus for the last few years, applying these particle engineering designs. And I just want to end by saying, we want to establish roots here. So I'm fairly new. I'm definitely not an expert on a lot of things. And so I'm really excited to meet people here to learn about really interesting animal tumor models, new analytical techniques. If you have ideas for different drugs that we could try, or if you have a drug that you want to try, or a different immune pathway, that's all really interesting to us. We love learning new things. So I just want to sort of, pass all of that information out to the ether. So in terms of acknowledgements, I'd like to thank my group. These are 6 all-stars. I'm really, really lucky to have amazing students like these in my group. So hopefully in a couple of years, you'll see a lot of work. You'll see these names a little bit more. I'd like to thank my collaborators and contributors to this work. Dr. Lisa Lee Sherrick has been instrumental in working with me and helping design some of these systems. So I'm really excited to work with her. Our Cancer Center members, or mentors rather, I have 3 outstanding mentors. I'd like to thank each of them for their time and advice. And I think that's it. So thank you for your attention and for your time, and thanks again for inviting me to come.
I'm going to moderate questions. Okay, since I'm going to moderate questions, I'm going to start. I almost forgot it when those balls were levitating, because that was a bit distracting, kind of unusual. So if you put interferon gamma into a cell, right, when the backpack gets phagocytosed, what happens to the macrophage? What is the result to the actual macrophage? Is that going to, is that hard on it? Does it stay alive?
So the question is, what happens to the macrophage if it internalizes interferon gamma effectively, or a particle containing interferon gamma? What we found is that interferon gamma is a very transient cytokine, and so it denatures really easily. If you put it in any sort of a solvent to try to formulate it into a particle, or if it gets internalized, the activity drops. And so, and also the other reason why we want interferon gamma to be released from the surface is that it can be released to other cells in that environment. And so if the particle is internalized, then that interferon gamma loses its ability to escape out into the environment and affect cells and its sort of neighboring cells. Yes. But in terms of, does it kill the cell that internalizes it? We haven't seen that so much, at least the cells, the macrophages we've been playing with, they're really robust. They can eat a lot of stuff, and they keep, kicking. So, but there might be a dose dependency. So it's a good question.
Yeah, cool. Oh, there's Mercedes. Mercedes, would you like to ask a question?
No, everyone's Can I ask a question?
Yes. Yes.
Okay. So with experiments, you compare interferon backpack alone or interferon. So I wonder what would happen if you could re-interfere an entity the backpack.
I think I heard, so I'll repeat your question, and you can tell me if I didn't get it right. So your question was, did you consider looking at a group essentially where you have a backpack bound to a cell, and then there's free interferon gamma just in the environment?
Yep.
We did not look at that at all. We didn't look at that, but effectively that's what we are generating, we think, because the release of interferon gamma from our backpacks is so rapid. Within 24 hours or so, 60% is released. So what we think is happening, at least in terms of the tumor microenvironment, is that after the course of maybe a day or 2 days, the backpacks are still on the surfaces, but most of that interferon gamma has been released at that point. So we haven't studied that explicitly. Was that your question?
It's just, yeah, in vitro, surprised with the effect that you see when you have the interferon in the backpack, but no interferon.
Couldn't quite understand.
Mercedes, you keep, your voice keeps disappearing in the middle of an important word.
I can hear it. Well, what I wonder is, in the experiment that it was shown, you have a huge effect with the interferon within the backpack, but no with interferon alone. So meaning there is some kind of synergism. And what I wonder if it's an effect of the anti-backpack itself together with interferon, because it's possible that it's interacting with CD44, like you say, and it's signaling through the CD44, because CD44 has an effect in macrophages.
I think I got the gist of your question. Let me see here. So I'm going to go back to this slide. This is in vitro data that we collected. I think this is what you're referring to.
Right.
So we did not look at backpacks with free interferon gamma in the environment. So I can't say what the effect of that would be. I suspect it would potentially be similar to what you see in blue here, with the combination of macrophages plus interferon gamma containing backpacks. One thing that is interesting to me, and maybe this partially addresses your question, is that if you look at just free interferon gamma and you look at the green curve, which is the blank backpacks, you see different responses compared to baseline. So what that tells us is that the backpack itself does have an effect. Interferon gamma obviously has an effect. And then when you add the two together, there seems to be some sort of synergy there. We don't really understand it, but there is something happening. I agree, it's very unexpected.
Yeah, that's why I said that it could be the backpack affecting the CD44.
I see your point. Maybe so. Yeah, maybe so. That's a great idea.
Okay, thank you.
We'll have to do a CD40 hyaluronic acid missing control. I like that.
James, can we have Eduardo go first? Okay, Eduardo.
Okay. So Wyatt, that was a great exciting stuff you have ongoing. I'm just wondering, can you use T cells in a similar method as delivery vehicles? And if so, what are the limitations to using T cells versus devices?
So Eduardo, can they hear your question?
Can I add to that? Because there's a question in the chat about dendritic cells. It's the same question about dendritic cells.
Okay, so Eduardo asked about the extendability of this work to T cells, and would there potentially be challenges? And then someone in the chat asked about dendritic cells, potentially for the same reason. So we've only looked at macrophages to date. I have an interest in looking at other cell types, but it just, these experiments take a lot of time, and so I think just with time and interest, I would love to explore more cells. Daryl Irvine at MIT has published a really interesting paper quite a few years ago now, maybe 8 years ago, where he has this malamide chemistry, where he makes these nanoparticles. I think they're lipid-based, they're multi-layer lipids, and he binds those to T cells. And he combines that with checkpoint inhibitors. And I think in that case, it worked quite beautifully. But I do think, checkpoints might be an issue that you have to think about if you're using T cells. Dendritic cells are really cool, maybe for a different reason, because dendritic cells are antigen-presenting cells, so maybe you could use this as a step toward a vaccine, like a cancer vaccine. So could you sort of engineer a backpack that could orchestrate some sort of antigen presentation, or could you use the backpack as a means to express antigens or display antigens on the actual body of the backpack? We've definitely thought about that. I don't know if that was the question in the chat, but we've thought about it. We've not done anything like it. We've just been looking at innate immune cells, arguably for the sake of simplicity. I don't know if maybe that is simpler, I'm not sure. But I think those are some really cool ideas for the future. I totally agree.
Ben? Oh, James, are you still there? Okay, Ben's going to ask while you're organizing.
So that was just incredibly interesting presentation. I appreciate you coming out and giving that in person. I work in ovarian cancer, and I thought to talk with you a little bit about using macrophages to treat ovarian cancer. One of the questions that I came up that I thought of, you mentioned you used the word macrophages become aggravated, that can't phagocytose backpack.
Frustrated, yeah.
Yeah, frustrated. Have you looked at how that changes the transcriptome of proteome?
I would love to do that. No, I think from a biophysical sort of perspective, there's some really interesting questions as to why. Oh, sorry, let me repeat the question. What was your name? Ben. Ben asks a really interesting question. Ben has an interest in ovarian cancer, and he asked about when these backpacks are bound to macrophages, macrophages have a hard time internalizing them. They get frustrated. And Ben's question was, have I looked at, or has anyone looked at like the genome, or looked at see what genes are being upregulated, maybe like a TAC-seq or RNA-seq, look at what's happening from a genetic or transcriptional perspective? The answer is we have not. I think from a biophysical perspective, we don't really know why these cells have such a hard time internalizing soft particles or shape anisotropic particles. We think it's because actin filaments have a hard time polymerizing around such an oblong shape, or in the case of a soft particle, because it's compressible, it has a hard time to deform the membrane and wrap around it. But that's purely anecdotal. We've never done any sort of genomic test or screen to actually look and see what's happening, and I would love to do something like that at some point.
So just to excel on that, you had mentioned interest in the new technologies, cutting-edge technologies. We just got on campus a new single-cell proteome analyzer. So it's basically detecting levels of different proteins and probably phosphorylated protein on a single-cell level. And so that
Cool. That would be really cool. I would love to check that out. That sounds, thanks, Eduardo. Yeah, Eduardo mentioned there's a really cool tool on campus that allows you to look at the secretome. Or is that right? Or
not only what's secreted, but also which is a proteome.
the proteome. Okay, I see. Thank you.
Are there any more questions from the Zoom world? Okay, one more in here.
Just a real quick general question. How hard is it to load your favorite drug in these nanoparticles? Is it pretty limited based on the specific molecule, or?
The question is, how difficult is it to load a drug into these particles? The answer is that it totally depends on the drug. Small molecules, generally speaking, are really easy. That's why in some of our future work, we're focusing on small molecules. They're so easy to formulate because they're hydrophobic, and most of these polymers are naturally hydrophobic, that they just go right in. If you start thinking about things like cytokines and antibodies, you have to preserve the structure of those proteins, and that becomes a little bit more of a challenge. That's why in the backpack project, we had to design that intermediate layer to facilitate that. There are ways to do it, and there's some really clever ways where you can put cytokines and antibodies inside of particles or on the surfaces. Arguably, the surfaces are even easier to modify and preserve sort of the structure of those drugs. But to answer your question, it really depends on the specific drug you're looking at.
Thank you.
Thanks.
Either way. Okay, if there are no more questions, thank you, Wyatt. Thank you for coming.
Thank you, Joe. Thank you.
We need to pipe in the